Hi, welcome to the webinar, and I hope everyone can hear me. If not, please sound out in the chat. Yes, and just a reminder that we will be recording this webinar, so and you are not able to show us your camera or speak in this chat, so please feel free to interact with us and ask questions in the chat at the end of the presentation. So welcome and good morning from the Denmark side. And the purpose of this webinar is to demonstrate how we at DHI work with data assimilation in the Mike Ecolab biogeochemical models. This webinar will focus on the data assimilation setup and results from the team as a user of the data assimilation modules and dataset instead of going into the theory behind the data assimilation itself. So presenting today is myself. I'm Xing Hui, and I've been in DHI for a number of years with experience ranging from Denmark, Singapore and Denmark through various marine assessments. And I focus a lot on water quality assessments and modeling. Right now, I'm involved in this project as a project manager integrating Copernicus marine datasets. And with me is Maj-Britt.
Yes, good morning. My name is Maj-Britt Kronborg. I've been with DHI for a number of years. Most of the time I've been working as a modeler, both hydrodynamic modeling, water quality, bacteria models, and biogeochemical models. Now I spend most of my time project managing various kinds of projects, environmental projects for Danish authorities, the Danish Environmental Protection Agency, and the Danish municipalities.
Yep, so I would also like to acknowledge the project team behind the data assimilation work presented here. So Rega, Anton, and Jose, who has supported us throughout this project. So DHI has presented the first webinar for the data assimilation back in 2024 on a similar topic that dove into the specifics of data assimilation. So just a quick recap, data assimilation is a technique that combines modeling and observations. It adjusts the models towards the measurements and brings the best of two worlds together. So the accuracy of the measurements and the higher spatial and temporal resolution of the models. So setting up the data assimilation in the biogeochemical model takes quite some knowledge on the biogeochemical processes in the system, the measurements, and the model, of course, the model. And then when we have used the best to our knowledge, we calibrate, we turn up and down the parameters and change and combine uncertainties in different ways and describe the uncertainties in different ways. Bottom line, it takes the knowledge of the knowledge on the system and quite some time to set up the data assimilation. So with that, we would move into our current webinar where we show some of the results and what are the data sets that we have been using so far. So first we'll touch upon, as I said, we will not dive deep into the theory behind. We will explain in short what is the data assimilation, how do we work with it in Mike modeling. We will go through the purpose of the project and overview of our systems, touch on the input data and the preparation of it. Then we will discuss the regional model data assimilation before the coastal local models. Finally, we get to the main focus of the talk, which is actually the presentation of the use cases in the inner Danish seas. We will present how these cases can enhance the biogeochemical results. After the presentation, we'll take questions. You have to write them in the chat and we'll take it from there. So this is the project system and overview. So why does this matter? Danish marine resources are under significant and growing pressure from offshore wind expansion, increased shipping activities, aquaculture, and so on. Yet today's marine monitoring is heavily relying on classical shipborne surveys, and the data and reporting generally takes a long time and sometimes with some delay. It's manual and expensive and spatially sparse. Therefore, with this integrated, the purpose of this project is to supplement and modernize the marine monitoring framework and also complying with the newer, the different EU directives written here and also the upcoming EU Ocean Pact. So the answer is what we call this digital integrated marine monitoring developed jointly by DHI and SCOUT, which is the Danish Agency for Green Transition and Aquatic Environment. The core idea is straightforward: to combine new observation technologies, satellites, autonomous buoys, ferry boats, drones, with mechanistic numerical models and machine learning to produce more and faster, better environmental information. So rather than replacing the traditional monitoring, we are supplementing it. We are trying to integrate it into the current modeling backbone and fill in the spatial and temporal gaps to give a good picture of the marine environment instead of just snapshots. So at the heart of it, I just want to emphasize the data assimilation workflow. It's a lot of information on it, but the key idea is the different phases. So the first phase is the model calibration and parameter testing. So together with Mike Ecolab, we have over historical periods conduct sensitivity tests, optimize parameters, and evaluate our models against observations. Then we have moved into the second phase where we do the hindcast with data assimilation, where we have run our calibrated model across the historical period with data assimilation activated and compare it against a baseline free run where we optimize and see what are the changes that data assimilation has brought. Then, namely for temperature and salinity, we have applied optimal interpolation, and for chlorophyll A and dissolved oxygen water quality parameters, we have used ensemble Kalman filter. All these would be elaborated later. So, and then subsequently, we are deploying it in operational near real-time system, where we will set up the operational system identical to what we have tested with integrating all the data sets that I've mentioned, and the DA corrected hindcast over a rolling window. This is delivered to Scout, which will be eventually shared. At least that's the target as well. So the key targets as part of this data assimilation is the improved summer chlorophyll A values, and also the oxygen depletion assessment, because every year there is oxygen depletion in Danish waters, and the public is pretty attentive to all these events and strengthening our understanding of the ecosystem. So what is data assimilation and how do we work with it? So in DHI, we work with mic models and modules. The mic 3 FM module for our hydrodynamics part, and then the Mike Ecolab module for the biogeochemical aspect, supplemented and coupled with the data assimilation module that we are using. So why do we use data assimilation? Because we are able to combine models and measurements as mentioned. We have full spatial and temporal description, process descriptions, and lower point-wise accuracy with models, but measurements, we have higher point-wise accuracy and limited spatial extent. So combining the two, we are able to resolve those. So what are some of the key model inputs? So we have the boundary data can be derived from global ocean physics. This is talking about the hydrodynamics models. We have satellite chlorophyll A data coming in from Sentinel images and then interpreted by Aarhus University. In situ observations from Copernicus Marine, shipborne measurements from NOVANA, the National Monitoring Program for Danish Seas. And then also marine underwater vegetation, like Copernic from Sentinel images. All these are just some of the highlights of inputs that we are using in our model. So what is also new to highlight here is that we have operational data for more than 400 sources from the Danish and global hydrological model run by the International Development Team, where we get freshwater flux and nutrient loads. We also have operational atmospheric nitrogen deposition delivered by Aarhus University. So, and the yearly initialization of the underwater vegetation with vision analysis and satellite images. So there's like a few different aspects that's delivered to the model, not just our data assimilation module. Oh yeah, and our meteorology is from the ECMWF. Yes. Okay. So the preparation of data. these are the types of data that we assimilate in the model, mainly time series, profiles, and satellites. There is a first check and the removal of data sets that are inconsistently uploaded, for example, in the Copernicus Marine in situ dataset, as it is planned to be operational. And then we will have a quality check on whether the in situ data is viable for use, for example, if there is too large of time gaps. And there is 6-hour averaging to smooth out noise. And for example, here, we are talking about for time series data, we have fluorescence measurements for surface chlorophyll, and there's a filtering of values during daylight hours with high quenching, and daily averages are computed before we assimilate them into the model. As for profiles, they are also very useful for us because we can understand what is the stratification in our model. So we assimilate them for the hydrodynamic parameters, temperature and salinity at every 3 meters. And but for oxygen and chlorophyll, we do have a focus on the bottom dissolved oxygen and surface chlorophyll. So all the other DA parameters will be discussed in the subsequent sections. Oh, and I have mentioned that the satellite image is also processed by Aarhus University before delivering it to us. So just look at the regional model before we go into the local model. So the area is the Baltic Sea. We carried out hydrodynamic model simulations, including data assimilation of salinity and temperatures. This is an overview of the station selected from Copernicus Marine in situ Baltic Sea dataset to supplement as a supplement to the Danish National Monitoring Program. This is an example of how the salinity stratification has been improved after we forced the model with bottom salinity has been improved after we have forced the model with the temperature, with the profile that we have obtained. The RSME has shown improvement at different depths and over time across the model. We also see that for temperature, this is a profile station, and we have also seen that for a time series station where we have assimilated salinity at the bottom and reduced the RMSE significantly. This also means that when we have a better stratification and understanding of the model, it's likely that we can also predict the biogeochemistry a little bit better. As for biogeochemistry, we also did the same thing where we have the model simulations of data assimilation, including the continuous buoy measurements from Copernicus, as well as supplemented by also the National Monitoring Program. This is also just an example that we have assimilated the bottom oxygen using the ensemble Kalman filter and achieves improvements in bringing our model closer to the measurement and reality. So why do we do this with our regional model? It's because we can downscale it from the regional and Baltic Sea. We would extract boundaries to feed it into our local seas and local bays and estuaries, and eventually achieve a higher accuracy and resolution in the very fine enclosed bays and estuaries, as well as fjords. So with that in mind, I will pass the time to Maj-Britt, who will talk about the use cases from the inner Danish seas.
Yes. Thank you, Singwei. So now it's time to zoom in on the inner Danish seas with 2 different use cases. And in both cases, we will be looking at model results from the Southern Belt Sea area, indicated here in the map. It's a very beautiful part of Denmark with a lot of recreational interests. We have here deep water that penetrates into the model domain via the Great Belt. Let me see, do we have a PowerPoint here? Here it is. I believe you can see it now. So we have deep water penetrating this way into the model area. And then on the other hand, up here, we have relatively well-mixed water that comes in via the very narrow strait of Little Belt here into the model area. And then we have the archipelago here south of the island Fynen, with shallower water, but also with a couple of narrow channels, which allows penetration of the deep water from the outside into the archipelago area. We've been running this model in Hindcast for a number of years without data assimilation. Now lately we have updated it with data assimilation, and we're also running it operationally now. The data we are assimilating into this model, I think Wei just told you about most of them, but just to repeat, so we are simulating ship-based measurements from the Danish National Monitoring Program, the parameters being oxygen, ammonia, phosphate, and nitrate. Then we are also using, and from these ship-based measurements are all sampled in the stations you can see in the map, where the yellow ones are the one we are simulating, the red one, the ones that we are using for validation. And then we're also using some continuous buoy measurements of bottom oxygen from two positions indicated by the arrows, one open water to the west and one in the archipelago sitting in one of these narrow channels. And then finally, we are using satellite-based chlorophyll data that is assimilated in all over in the domain whenever data is available. And then finally, I should mention that the biogeochemical model results are based on a hydrodynamic model simulation, where we're also including data simulation of salinity and temperature from these stations indicated here. The first use case we'll look into is focuses on the surface chlorophyll. In the model results we are going to look at, we are simulating satellite-based chlorophyll data, and these data are basically sentinel-free data, which has then been processed and optimized to the Danish conditions by Aarhus University here in Denmark. Satellite data is a good source of information in general, and it's also good for assimilation due to the 2D coverage, but we also have to remember that we do not get a perfect picture with full coverage every day from this data source. Some days are cloudy, we only get half, other days are even worse, and sometimes we don't get any data at all. So if we count the number of days during a season lasting from 1st of March to 1st of November, we get around 60 to 70 days with satellite data in the open areas of the Southern Belt Sea area. Here we're looking at two examples of what it looks like when we assimilate satellite chlorophyll. Both stations are indicated on the maps, are open water stations. Blue is surface values, light blue is the baseline, dark blue is the assimilated results, and red is the bottom results. The stars indicates satellite observations, which we are simulating into the model, whereas the triangles are the ship-based measurements, and they are only here for reference or validation. And we clearly see that there is no data before 1st of March and relatively few in March and April, which is kind of sad, since then the satellite missed to detect the spring bloom in the area, and then it's also not an information that we can carry on into the model. But for May, it picks up with more data, and we see that we succeed in adjusting the chlorophyll concentrations to a higher level in general during summer and fall at these two stations. Here we have an example where we have zoomed in on a specific day where we have a good satellite coverage. That's the picture to the top right. It's the 9th of September. To the left, we see the model results for that day at the top for the baseline and the bottom for the data simulated results. And we clearly see a change from baseline to DA results, so the data simulated results. And when we estimate the difference, that's the picture to the bottom to the right, we see that the concentrations has been adjusted upwards almost all over the domain with something like 0.5 to 4 micrograms per liter. When we compare the DA results directly with the observation, we see there's an overall good accordance with the observation, but we also have to remember that this is not only a result of assimilating this particular satellite picture into the models. It's a result of it's an integrated result of assimilation of satellite observation of days to weeks before this time. The second use case we will look at is bottom oxygen in the area. in the area of the southern Belt Sea, it's often strongly influenced by oxygen depletion during summer and during fall. It means, of course, unhealthy ecological conditions for both fauna and fish. And it is, like Singwei said, something that is a lot of concern of here in Denmark and get a lot of attention in the Danish media. So it's something that people want to follow, and especially, of course, the authorities. The figure shows model results, including data assimilation of bottom oxygen for a 2-week period in August 24. And the red is shows us the severe oxygen depletion with concentrations below 2 milligrams per liter. So as you can see, it's a widely spread phenomenon, not only in the deep water, but also in some of the fjords and in the archipelago. To show you what it looks like when we assimilate and how much we can adjust the model, we here have an example of data assimilation at the bottom oxygen at a relatively deep station indicated on the map. Blue is again surface conditions, model results, whereas the red is the bottom conditions. The pale red is baseline, the true red is the assimilated results. At this station, we have both continuous data and ship-based data to assimilate in. The continuous data is here indicated with the purple dots, and the ship-based are the red triangles. We only have the continuous data for 5-6 periods, but fortunately, they are available right when the model needs some help, and we can see that we get the concentration lower quite nicely relative to the baseline run. Outside the period of the continuous data, we assimilate towards the ship-based measurements, and even though they are not as frequent, we see that we get quite good effect of the data assimilation anyway. We can also notice from these results that we do not get the model results adjusted all the way down to the measurements. We can get it down all the way down to the measurements by assimilating stronger, but then we very often get instabilities in the nutrients. So a major part of setting up the data assimilation routines in the biogeochemical model have been to find the right balances of how strongly and how to assimilate the different parameters all together without introducing instabilities in the biogeochemical balances. Yeah. Before we just looked at data assimilation at a single station, and here we'll have a look at the spatial extent of the data assimilation, or the effect of the data assimilation. The figure to the left is average bottom oxygen for September in 2022, and the figure to the right is the same for the data assimilated model results. And by eye, we see a clear difference, but estimating the difference shows a very clear lowering of the average oxygen concentrations of 0.5 to 2 milligrams per liter in the central open part of the southern Belt Sea, and likewise in the archipelago. And please remember here that most of the station, we only have measurements every 2 weeks, or even less frequent than that. And yet we get a widespread spatial effect of assimilating these data into the area. And we can even see it in this strongly integrated measure as monthly mean is. The question is then, what can we use it for, and what can the authorities use it for? The obvious answer is that we can supplement the existing observation-based monitoring with model-based data at a higher and spatial and temporal resolution. The picture to the left is an example of the existing observation-based maps of bottom oxygen, which the authorities has available today. Every month during the summer, a new map is issued based on the station distribution that we looked at before, and then a qualified interpolation is applied between the stations. With the model data, we will be able to supplement this information at a much higher frequency and covering the full area. Here an example is given with weekly maps of bottom oxygen showing the depletion or the development of the depletion, which will be something that can be difficult to follow if we only have monthly maps. Another measure that we have been suggesting to the authorities is a measure of how big an area is affected by the oxygen depletion. The areal estimate could be given for the whole area or a subdomain or sub-area. Here an example is given where we are looking at the extent of oxygen depletion in the archipelago over time. This reveals that the area of oxygen depletion is not something that builds up and stays at a certain level and then declines again in fall. Instead, it builds up, declines a number of times over the summer. And probably in this area, resembling the wind conditions, where strong wind winds refreshes the not too deep bottom layers in the region, and calm periods of winds serves to strengthen the oxygen depletion. Finally, it might also be relevant to discuss duration of oxygen depletion events, since some species are able to survive shorter periods of low or no oxygen available. And here is an example given on a bit longer time scale, showing the number of days with moderate oxygen depletion in July 24, indicating the persistence of the phenomena. So that was a couple of cases of what we've been suggesting to the authorities on how they can work with these model data, and the fact that we have a much higher spatial and temporal resolution of the information. And with this, I will hand over to Singwei again to wrap up.
Yep. So thank you, Mybrit. Yeah, so with that, we have seen that it's a generally good effect for assimilating oxygen and chlorophyll measurements. The assimilation has a widespread effect in the South Funan archipelago and in the southern Little Belt, and it's not just locally around the assimilation stations. So one of the outcomes of these exercise is the maturing of our operational system for inner Danish seas for integration into our national and European data sets. And we are continuing discussions with Scout and creating what kind of outputs will come from the model and what will benefit users the most at the end of the day. So there's supposed to be a small little animation here, but it's not playing. So hope that's fine. Yeah. Oh, okay. There we go. Yeah. So yeah, thank you for your attention. And it's time for questions.
So we have a question here. Thank you for your presentation. What is the hot start time before actual data assimilation? I'm not sure. Maybe we can ask the one posting the question to elaborate a little bit. I'm a bit uncertain about what it is.
So we did our hindcast in 2024, where we have initial conditions from our previous model runs.
Yes, without data assimilation.
Without data assimilation, if that's answering the question. And then when we run it operationally, it's going to be continuous.
It is, and it'll be very interesting to see if that changes the quality of the results, because there is, of course, an effect of the initial data not being data assimilated. Likewise, some of the model results that we have been presenting today has also using boundary data, which is not assimilated. But of course, again, in the operational setup, everything will be tied together, and initial data as well as boundary data will be data assimilated data as well.
And there's a second question here. Briefly, can you explain how the model is adjusted each time data is assimilated? Thanks. I think maybe I'll give it a go before Mybrit supplements. Mybrit also mentioned about how we adjust the models. Usually it's by defining some kind of standard deviation and defining how much uncertainty you want to place in a model and versus the measurement. And the model will be able to assimilate towards that. So you can adjust the assimilation strength in a way. So that's for the data assimilation stations.
Yeah, and then we can also locally for each station or each type of data set a set of parameters that sort of determines how far away we let the model see the measurement. That can be done spatially, horizontally and vertically, but you can also do it in time. And that's why we can get like a pretty good effect, even though we only have less frequent data, as we just saw it in the example with the oxygen data. Even though we only have data every 2 weeks, we let the model see the data some days before it actually gets to the measuring time of measurement, and also some days after. And that helps to prolong the effect of the measurement in the assimilation. But we should of course always consider for how long time and how far away the measures are representative in the water bodies.
And just a follow up as well, what means baseline? Is it hindcast simulation?
Yes,
well,
in this
in
case,
this
baseline
case.
means it means everything we've shown here is hindcast simulations, but in this case, baseline means simulations without data assimilation. So a baseline to show the effect of the data assimilation.
So are you using the model to examine ways you could reduce the phenomenon of depleted oxygen near bed, i.e. with effect of reducing overall nutrient loads to the region?
Yeah, I mean, that has things like that has been studied very thoroughly with these types of models. It's not something we have studied yet with data assimilation included, but it could definitely be something that we will look into over the years.
Yeah, so as mentioned, the key targets of this exercise was the focus on summer chlorophyll A and oxygen depletion as the main two use cases, but it's a continued effort where we will continue trying to consider all these other aspects. And their next question is somewhat related. So what is the limitation on data assimilation? How many WQ parameters can be data assimilated at once? In theory, there is no limit except you can assimilate all the parameters. However, as Mai-Britt mentioned, there is limitations on how much instabilities you can cause in the model if you try to assimilate too many parameters at once. So maybe, yeah.
Yeah, and also, I mean, running ensemble Kalman filter data assimilation, it's quite heavy. We run these rather big models with 10 ensembles only, and that's quite an effort to do that. So just in terms of hardware, there's definitely, we also see that there are limits to what we can do. Otherwise, as Singwei mentioned, I mean, if questions pop up later, please just contact us. I think it's getting silent here in the chat. People saying thank you, thanks a lot.
So thank you. It was good to present this project on behalf of the project team, and have a good day ahead.
Thank you. Bye-bye.
Bye-bye.